The definition Miller in 1930 had defined the transpiration as the loss of water in the form of water vapors from the stem and leaves of the plants is called transpiration. We see types of transpiration. The first is lenticular transpiration. Some water may be lost by woody stems through lenticels, which is called as lenticular transpiration. Number 2, cuticular transpiration. Cuticle is the layer of waxy substances present on the outer wall of the epidermal cells in the leaves. This cuticle is in direct contact with the surrounding atmosphere. This thickness of cuticle varies in different plants and generally in xerophyte it is thick, in mesophyte it is thin and in hydrophytes it is absent. When cuticle is thin due to effect of surrounding atmosphere, some cracks develop in this layer due to direct contact with atmosphere. Through the cracks in the cuticle, some water from epidermal cells diffuses in surrounding atmosphere in the form of water vapors. Such loss of water in vapor form through the cuticle is called cuticular transpiration. In leaves with thin cuticle under dry condition, when stomata is closed, cuticular transpiration becomes more significant. Out of total loss of water from the plant body, only 5 to 10% is due to cuticular transpiration. Third one, stomatal transpiration. Stomata are minute pores present in the epidermis of aerial organs of a plant. They are surrounded by specialized epidermal cells called guard cells. The guard cells exhibit movements and control the rate of transpiration. Maximum amount of water lost from the plant body through stomatal pore. The loss of water through stomata is called stomatal transpiration. Such aerial leaves are with maximum number of stomata, about 80 to 90% of total loss of water from the plant by stomatal transpiration. Out of two types of foliar transpiration, stomatal transpiration is very important for addition of water vapors in the surrounding atmosphere. Now we see mechanism of stomatal transpiration. So this is the cross sections of leaves. It is the structure of stomata and this is how stomata they are arranged in the leaves. Now the cross section of leaves show presence of stomata in the epidermal layer. Each stoma or stomata shows an opening or pore guarded by two special epidermal cells called guard cells, and these guard cells again surrounded by the epidermal cells which are called as subsidiary cells. Just below stomata in the subepidermal region or mesophyll region, a small space is present known as substomatal cavity or substomatal space. From this substomatal cavity, water diffuses in the atmosphere when stoma is open. Now we see the mechanism of the opening and closing of stomata or stomatal transpiration. This is the diagram which show how stomata they are arranged in leaves, and the mainly this substomatal cavity through which water loss takes place. So the diffusion of water from the moist cell walls of turgid mesophyll cells into the intercellular spaces and substomatal cavity. The diffusion of water in vapor form from the substomatal cavity into surrounding atmosphere when stoma is open. Diffusion of water vapors from substomatal cavity through stoma into atmosphere is called stomatal transpiration. The stomatal transpiration takes place when stoma opens. It does not take place when stoma is closed. Now we see the opening and closing of stomata. So this is the structure of stomata which show the stomatal pore. It is surrounded by guard cells which is surrounded by epidermal cells, and this is the diagram which show how the conditions when stoma is open, and the next shows the stoma is closed. The opening and closing of stomatal pore is due to the movement of guard cells. Guard cells are the special types of epidermal cells because they show the mainly different structures. Guard cells are kidney shaped in dicot and dumbbell shaped in monocot, that is like grasses. Also they show difference in their wall thickness. In normal epidermal cells, all walls are of uniform thickness, while in guard cells, the inner walls they are thick and outer wall is thin. This difference in the thickness of wall is responsible for the movement in guard cells as well as for stomatal movement. When stoma are open, the guard cells are flaccid, and stoma are closed, guard cells are flaccid. Now we see the how opening and closing of stomata takes place. When guard cells absorb water, their turgidity increases. The increase in turgidity increases the pressure on the wall. That is, wall pressure increases. This wall pressure acts in outer direction. Due to increase in the wall pressure in outward direction, the outer thin wall bends and becomes convex. Such bending of outer thin wall pulls the inner thick wall in the outward direction. This pulling is responsible for the movement of two inner thick walls of the two guard cells away from each other. When two inner walls of the guard cells move away from each other, the space surrounding the pore widens and stoma opens. Now, when we see when the guard cell lose water, the inward pressure is created by the outward walls on inner thick walls, due to which two inner thick walls of the guard cells move close to each other. When two inner walls come very close to each other, the stomatal pore closes or stoma closes. This shows that the opening and closing of stoma turgor in the two guard cell is responsible. In this entire mechanism, of stomatal transpiration, the factors responsible for changes in the turgidity in guard cells, they are not mentioned because they are not correctly known. Thank you.